All right, and we will turn it over to Trisha.
Can you still hear me, Megan?
I can still hear you, Trisha.
Okay. Well, I wanted to first apologize. I felt so bad because when we tried it, everything worked. So I felt really awful about last week and having the technical difficulties. So I'm sorry again. And I hope you guys all had a really good St. Patrick's Day so far.
And you guys can, if it doesn't already show it this way for you, you can select speaker view on the top right. And that way it'll make Trisha nice and big, and then we can see her pretty face in the corner too.
Oh gosh. Okay. How are you guys tonight? Good? Okay. If do you guys all have your hot water bath or hot water ready and ice cold alcohol ready? So Megan, since I kind of missed a little bit last week, is the best option here, like, is it just running through the directions? Does everybody have the direction sheets out from the binder?
Yeah, and I feel like a horrible host because I don't actually have a copy of the binder with the page numbers on it to tell you guys what page it's on. But they are numbered.
It's on page 9.
Awesome. Okay.
Yes.
That was it. I said page 9 in the email about equipment, and that was a guess. So it was a good guess.
Yeah, it is on page 9. And so do any of you guys actually do DNA extraction currently?
We haven't much.
Okay, so maybe 2 of you? 2 or so? Okay. So do you guys understand the basics behind DNA extraction, or why we're going to be using these different chemicals like the soap and the meat tenderizer? Okay, so for DNA wheat extraction, if you guys want, we can go ahead and get the stuff started, since it has to sit for 15 minutes. And then during that time, we can talk about the different materials that we add and why we add them and how it helps us actually extract the DNA. Are you guys okay with that?
Yep.
So you guys want to take, a couple minutes to get your wheat germ, your meat tenderizer, your hot water, soap, and stir it. Couple minutes. I'm going to get mine too.
And then, yeah, feel free to ask questions, unmute to do that, or just throw them in the chat. I'm keeping an eye on that as well.
Thank you, Megan.
No problem.
So my wheat germ has been soaking, and I've got it like a smaller beaker inside of a larger beaker for a water bath. So I just take that out now to add the tenderizer and the shampoo.
So I did the experiment earlier, and I actually had put everything in with the hot water and the soap to make sure it worked. So I had the meat tenderizer, the wheat germ, and the soap, and then I poured the hot water in and mixed it and let it sit for 15 minutes.
It doesn't need to be in a hot water bath now, right?
It does not. It worked earlier without. So in the directions that Megan and Kristen sent, it did say to use all of the meat tenderizer that she sent, that Megan put in meat tenderizer and wheat germ, and then heat the water, and then add, it was about a teaspoon, a squirt.
Yeah,
of
squirt.
the shampoo. And then shake it well and let it sit for 15 minutes.
Is it okay if the wheat germ sits in hot water for longer than 15 minutes? Yes.
So I'll give you guys a few minutes to get your stuff. Do you have any questions about DNA extraction right now? Okay. I guess, Megan, we just kind of wait and see when everyone's done.
Yeah.
Okay.
So I see some stirring and
Oh yeah,
mixing. Yeah, I see yours swirling too. Nice.
I am.
Yeah, and again, going back to the question about it waiting 15 minutes, I would say 15 minutes is probably a minimum. So if it sits longer, that's fine. And yeah, I would say maybe if you were doing this with your students, you could start it a little bit earlier and just leave it. And then add the soap and tenderizer and give it the last 15 minutes.
I also recommend making sure your meat tenderizer is not expired in 2018. Apparently that doesn't help with the DNA extraction process.
Okay.
Found that out today, so.
Oh. Well, and we bought some on Amazon like a month ago or right before we sent this, so it should not be expired.
Yeah, well, I didn't want to use your resources today when I ran through my trial, and my trial failed.
Oh.
And my trial failed again. And then finally I used your stuff and it worked. So, and then I started troubleshooting. I probably ran 7 wheat germ DNA extractions throughout the day, trying to figure out what part of my stuff wasn't working right.
Oh no.
So it was good failing and trial and error. So I think it came down to maybe old wheat and old meat tenderizer. So.
Well, and then the meat tenderizer that you guys are using is the papaya based one. It's papayan or whatever. They make bromelain too, which is the pineapple based one. But yeah, I got the papaya one.
Hey, you might have told us this already, but could you tell us what the tenderizer does specifically, and then what the soap does? Does the tenderizer do the membrane?
Yeah, so is everybody ready for that while we're waiting?
Oh, if you're not, yeah. If you want to wait, that's fine too. I just want to make sure I knew what did what eventually.
Yes, we will.
Okay, no hurry.
Okay, I just wanted to make sure everyone was done. I mean, for the people that I can see, it looks like you guys are ready. So, guys, you can extract DNA from almost anything in the classroom. Strawberries are really popular. Bananas are really popular. I had students actually use one of the many roaches that are in our school and they extract their DNA successfully. Kind of gross, but you know, and obviously cheek cells, but with COVID, I probably wouldn't go there.
We actually have some success with frozen strawberries. So like if the strawberries are an arm and a leg, like they are sometimes, you can just buy a bag of frozen strawberries and it works. Just let them defrost.
Yeah, I mean, it's pretty much universal. I mean, you can do, you can even set up, depending on how much time you have, you can set up a DNA extraction lab where they actually use different chemical or different things like different meat tenderizers or Epsom salts, regular salt, different soap, heating. You can do stuff with lima beans. And if you like let the lima beans sit in water, then you're actually getting the bacteria, and you can extract the bacteria DNA. You can extract plant DNA. You can extract animal DNA from like liver, like meat. So if you kind of wanted to compare how you have to break down different cells, you can set up labs and have them kind of experiment with that. But the soap.
I'm sorry, I'm interrupting you, but I'm excited.
No, go ahead.
Can I ask you about the bacteria thing? Like, so just like a cup of beans, dried beans, I'm assuming, let them soak overnight. And then like with minimal amount of water, like, do you have to do anything to concentrate the bacteria? Like, can you tell me?
Yeah, so the, and I can actually send you the lab. So my biotech class, one of our intro labs is this just very like stuff from home DNA extraction stuff. And they extract from onions, they extract from wheat germ. They do the lima bean bacteria, which is lima beans, soap, fresh papaya juice, which I struggle to find, salt, 2 different kinds of salts, and then some different buffer and aspirin. And then the lab that I have goes through the different reasons that you would add different chemicals for different kinds of organisms. So like in here, in this lab, the salt, the soap is, if you think about the cell membrane, what is the cell membrane made up of? Anybody?
Yeah, phospholipids.
Phospholipid bilayer, right? So the lipids, you know, have hydrophilic and hydrophobic properties. And if you think about just like greasy, fatty, yucky pans, what do we use to break apart, you know, a dirty lasagna dish? Is we do the soap. So the soap helps kind of break down the lipid membrane and the nuclear membrane, which allows the stuff to be able to leave the cell. So the soap helps break down the cell membrane, and then the also the nuclear membrane. And then the meat tenderizer, like Megan said, has the papain or whatever in it, which is actually a protease enzyme, which helps like kind of cut the proteins away from the DNA. You know, in the nucleus, the DNA is wound around proteins and coiled up, and the protease actually helps detach those proteins from the DNA. And this, I don't know, Megan, did you use, like, I just have the Kroger.
Yeah, it's really similar. I mean, I can find it on Amazon. It was a big old bag of it.
Yeah, this has salt, sugar, and it has the papain in it, like what you were saying. And adding salt to some of these DNA extractions, the sodium and the chloride ions helps kind of neutralize the negative charge that DNA has. And so it's not like forming bonds with other proteins and things as well. So that's kind of, I think, the 3 main things that go in most DNA extractions is for sure soap and salt. You know, we did bananas with kindergartners and baggies, and we were just mixing haphazardly soap, salt, and banana, and we're able to extract DNA every time.
Like, so much fun.
it was very fun when the kindergartners came to our classes last year. I was cleaning for days, so. But they loved it. I mean, they don't know really what DNA is, but the kids like talking to them and showing them, and they get to mix things and put the stuff in test tubes. And then the last thing, which we'll get to here in a couple minutes, is the alcohol, which is, it just helps isolate that DNA like up into the top layer. It kind of precipitates the DNA. And so it's separated from the proteins, and it's mostly just DNA that you guys will end up with in like a white cotton ball looking wad of DNA, I guess. I don't have a good scientific term for it, but so that is kind of I guess the four main things to have for DNA extraction. And like Stephanie said, a lot of things work. So, and wheat germ, you guys can you buy a box and keep it for, I don't know, because the one
expires.
Oh, actually the one I used is not expired. The other one I used that worked was. So I don't know. I have to go back to looking at one worked and one didn't.
So what's different? What's the difference between the meat tenderizer and the contact lens solution? Because I've never done it with meat tenderizer. I've always done it with contact lens solution.
I have never done it with contact lens solution.
Oh, okay.
So
I did the strawberry one with just regular salt, and then I bought a giant thing of like Gain laundry detergent and would just take like a couple of drops of that and put it in water.
Okay. All right. Well, salt's cheaper than contact lens solution, and and not as dangerous.
Right. So Will, did that help answer your questions about the different solutions that you put in or different chemicals?
So it's the salt that keeps DNA's negative charge from bonding to other proteins. That's what you said?
Right. It's the sodium ion. You know, when you have salt and it breaks apart, the Na+ will help kind of neutralize that negative phosphate group on the DNA. Or I'm sorry, was that you, Thomas, that asked the question?
Yeah, it was me, but good job. I'm sure Will had the same questions.
Okay.
Yeah, it was my question too. Thanks for answering it. Yeah.
Okay. So I don't know if you guys have any other questions about it. I'm really anxious to see what you guys your DNA. But I don't think it's quite been 15 minutes. I failed to set a timer.
We should be there about 5:30.
Okay.
Halfway
there. So is there anything else about DNA or other DNA extraction methods that you guys have questions about? I mean, I don't know.
You said you had like a write-up that kind of talked about all the components that you share with your students. Is that something that you have shared on the Google Live that we can access?
I will email. I think Megan and Kristen actually have it, but I'll give it to them and they can get it to everybody.
Okay, thank you.
You're welcome.
Yeah, is that Mark's big one?
Yes.
Okay. Yeah, I have that.
Yes.
I will put it in the folder.
Yes, that's the one. I don't think we made any changes, but I can make sure to send you at least the newest version.
Awesome.
So Yes, so I just saw in the chat food coloring to dye the alcohol when the students extract DNA from their saliva. Another thing that we've done is using like different color Gatorades to actually swish your cheek cells around. You can use like Powerade, blue Powerade is super fun. Stuff like that helps. And then I know in the directions it talks about making the necklace and things like that they like if they get to do their own DNA. Are any of you guys more like, do any of you guys do other labs that would be more in depth than just like regular bio or honors bio for DNA extraction? No? Okay. I was just asking. We've been doing DNA extractions from organisms in our little like wetland area. So that's been pretty fun. Dragonflies, leeches, you know, the whole bit. So.
Well, then the DNA Learning Center in New York has a program for isolating it from ants. So they're doing an ant barcoding project. So you isolate the DNA and then run it and basically get a sequence through PCR and then get that into their database. So that's a fun project. I don't know if they'll do virtual again this summer, but last summer they did virtual kind of hands-on labs with that.
That's really cool. I think I wrote that down when we talked.
Megan, speaking of PCR, is that something that teachers can borrow at the equipment depot?
Yep, so we have a couple thermal cyclers and we're working to get some more funding to get more, but we can loan those out to your class. We really love mini PCR and they have a mini 16, which holds usually 4 student sets of tubes of PCR because it's usually 4 tubes a piece.
Okay.
So yeah, happy to share those.
When you loan that stuff out, Megan, do you have all those reagents that go with that stuff, or would that be something a school has to get?
That would be separate unless we're doing that as an implementation. So hopefully we'll be able to do another round of our Going Viral, which we did PCR for bacterial and viral disease detection in that. So if that were the case, you could do the same implementation where we send you a whole set of supplies to use for your class. But for right now, yeah, I can loan you the equipment and then you would just need to purchase the reagents. I can recommend some and get you a discount with mini PCR. So yeah.
Yeah, and I've generally found like those kits are actually fairly inexpensive. Like for mini PCR, I mean, they're not very expensive for a class. And, you know, if you're really conservative, you could probably like split it and cover 2 classes for $30 a piece.
Yeah.
You guys have any other questions, or has anyone else tried anything for DNA extraction? I know regular biology, honors biology, even environmental science, any of this stuff is so easy to do and the kids love it. One of the other things that I've had kids do is actually take the stringy DNA and put it on microscope slides, and they actually make their own wet mount, and then they try to look at it under the microscope. Sometimes I feel like they're a little disappointed that they can't see that it's like double helix and they can't see the A's, T's, C's, and G's and stuff in the DNA. They think they can, but it's also really good for them to just use the microscope and get that extra use. So are you guys ready to take your pipette? And
Saw some pipettes.
I'm gonna have to go get my cold alcohol. In the directions, I know that it says you might, like when you put it in your little tube that you guys can shake it. Sometimes too, just letting the cold alcohol run down the side, it makes a really nice layered like alcohol on top, and then the DNA extraction solution on the bottom, and the DNA will just kind of precipitate up. So you can try both. Okay, guys, have you poured your alcohol down?
No, not yet.
Okay. I don't know if you can see what I'm doing. I have it. Yeah.
You got a test tube. They have the micro tubes.
Yeah, and it's because I had to use everything earlier, and I was also very worried that something was going wrong. So I actually mixed everything in just your one tube, and the concentration of DNA, like the stuff that came out of it was a lot more than typical. So if you ever want to like play around with it and lower the amount of water that you add, it might help. It worked great for mine earlier, one of my trials.
Nice.
And I don't know
This
if I
doesn't
have.
work. I'm going to be so disappointed.
Well, I was running about 50% through the day between everything that was expired and old and getting sidetracked with my classes. So
Yeah, I bet you couldn't forget those, could you?
so I don't know if you guys can see in this one, there's the DNA, like the white stuff starting to pull up with some of the bubbles. Can you guys see that?
You're really
Trisha,
close.
you can maybe dial the focus on your camera a little bit.
Okay.
Here, does that
I'm
help?
so hesitant to touch
Yeah.
anything.
Oh yeah, Stephanie's got a blob. DNA
Yes.
blob in the alcohol.
Yeah. And so I kind of did what Trisha did. I just kind of dumped it all together, and then in a bowl, and then I poured some back in here and added the alcohol to it, put the cap on it, and just let it sit. And that's what it did.
Yeah, let me see. I have such a weird glare on my desk.
Yeah, hiding from the light.
Yeah. I don't know if you guys can see that right there. It's a big white blob. But maybe I can go like this.
Yeah, I see the blob that way.
There it is. And then there's this stuff that's kind of climbing up into the alcohol layer. So after that round, I'm shooting over 50% success, which is great.
It's like it's kind of looks like snot. Is that what I'm looking for?
Yes.
I've got this
Yes.
stuff that keeps doing this and the
Yeah, it's really neat. Sometimes when the bubbles, like if the bubbles kind of draw it up, like it looks like the Milky Way, like the bubbles are stars, and
Yes.
yeah, it looks really pretty. So
I don't teach biology or biotechnology. I just teach chemistry, like regular chemistry. So when I set this up, set up the labs for like last Wednesday and this Wednesday, my kids are all like, "What are you doing? Can we do this tomorrow?" I'm like, I got to try it first. So yes, I think we might do this tomorrow. They're excited about this.
it's really neat because you can talk about the negative and positive charges of things, and so
The enzymes and the catalysts and that kind of stuff. Yeah, bring that stuff
exactly.
in.
Exactly.
All right, so how do I get it out of the tube? I pour it out.
So sometimes you can actually, like if it's a big enough, you can kind of suck it back up with the pipette and then put it in like just straight alcohol. If you have any like little plastic loops, or like I have some inoculating loops, but not everybody has that. You can kind of wind it around. Sometimes a glass stirring rod, you can wind it around, and then you can stick it in a tube that actually just has alcohol, and it'll stay in there.
And with the plastic pipettes, if the tip is too small, you can cut the tip off and then use it kind of further up to have like a bigger opening to pull it in.
It has to stay in alcohol. Is that right? Or like they couldn't pour it out on the table to see it.
I mean, they could, like I said, my kit, they could take it apart with tweezers or whatever they wanted to do. And, my kids look at it under a microscope. So, I mean, really, I think that's some of the neat stuff, is seeing what they want to, look at afterwards, or what other questions they have afterwards. You can do different, so certain things like for gel electrophoresis, like what the guy was telling us today, and Matt, what he was saying. So there's a little stain called Fast Blast, and it actually would stain DNA like a blue color. It's really neat, and then they can look at it. So you could even take an onion cell and put some Fast Blast on it, look at it under a microscope, and it would stain the DNA, and then you could take the same onion and actually extract the DNA, and then they could see it out of the cell. And I think it's always alarming, like some of the bananas and strawberries and things, and even what Stephanie had such a huge amount of DNA. I think they're surprised by how big of an amount it is, but then when you start talking about how small cells are and how big, how many base pairs DNA is, that when it all comes out of the cell and makes such a large, sample of it, it's pretty eye-opening for them. And then another thing about DNA that you can do is, like, if you take like yarn, if you take 2 pieces of yarn and a drill, and like do the drill in reverse. I don't know if it has to be in reverse or forward, I forget. But you can take the yarn and stretch it out to the end of your room, and when you turn the drill, if someone's hanging on to the end of the yarn, the drill will coil it up into the smallest little ball. And so that can also kind of show like how big DNA is, and then how compact it can be in the nucleus, like reverse.
So I'm looking at the chunky stuff, is that right?
Yes.
Chunky. Okay. One of my older well, he wasn't old, but like about 8 years ago, he did it with strawberries, and I remember him twirling it up the last stirring rod. So these little chunkies are kind of different.
Yep, and it's nice because it floats up into the alcohol. And then you can grab it out of there. So I don't know what other questions you have or ideas that you guys have. Think.
So someone in the chat asked, what's the importance of the cold alcohol? Why has it got to be cold?
You know, I think it works when it's not super cold too, but for some reason, the cold layer, let's see.
Yeah, I think it's like the temperature difference. It can like
Helped a college kid with this question, and their professor said that it slows down the motion of the particles to help everything happen without it reattaching. Something like that.
Yeah, like reattaching to any protein or cellular and stuff. Yes, it's perfect.
Yeah, and then Janet, one of our other teachers who does the wheat germ pretty frequently, said that she'll use other alcohols for this. So like if you have ethanol or something sitting around, they don't have to be as cold, but it works best if you're using isopropyl to do it super cold.
And this stuff was 30%. The bottle I have is 30%, so it doesn't even have to be.
I sent you 70. But yeah, you don't need the 91 or 99 to do it.
Sorry. I transposed my numbers again. Yeah, that's what I was gonna say. You don't have to buy the name brand fancy stuff. Just the off-brand works.
No, we ended up with the stuff from Walmart. I ordered from Amazon and it got delivered in a Walmart bag, so.
Love it.
Yeah, we're just starting to see alcohol back on the shelves around here. It's crazy.
We just needed some to clean the depots, so I was having to scour Publix. Every time I walked in there, I went and checked the alcohol.
I mentioned it in class and somebody drove, like one of the upperclassmen drove like 30 minutes to a neighboring county.
Wow.
And found some. And it was like 2 bigger jugs of 91. And I was like, this is a jackpot. So
I did see that at Ace Hardware the other day. So yeah, the big old jugs.
Yeah, we blow through some alcohol wiping off our desks and things around here. So, but I don't know, DNA extraction, it really, like, everything spans the gamut for courses on stuff that you can bring up. And, I know certain classes have strict timelines and everybody always feels very pressured to finish curriculum and content. But if you have time to just even have them research things or just lay stuff out on a table and see what they can mix up and see what's successful and not successful, it's really neat to see. And they feel really awesome when they get it. So
that would be the explore and elaborate part of your 5E, right? And that's really the heart of the 3D learning in Georgia now since 2016, is putting the science back in the kids' hands and giving them the ability to discover and explore. But I'm so with you on how important that is for students to be able to do that. And I know Tom, he's probably shaking his head, yep, right now, because it really is about getting teachers to have experiments like that for their students and that kind of a learning. So I'm glad you mentioned that.
Yeah, because you can talk about what works and doesn't work. And even if the students use different amounts and get the same result, I mean, there's just endless things that you can talk about and compare, and even like data, like how much did you get, having them quantify stuff. And I mean, you can really turn it into a really good experience and inquiry kind of based activity if you have the time.
Yeah, we've just started DNA in my classroom, and I'm like thinking, where's my drill? Because I'm absolutely bringing my drill to class with me tomorrow, going, all right, guys, let's see what happens. I heard about it. Let's try it.
It's really, it's hard. I think things like that are important because it's hard for them to visualize DNA in a cell and how it coils. And when you talk about mitosis and meiosis and everything, it's just eye-opening for them, and they remember and they enjoy it. So. I don't know if you guys have any other questions.
So,
oh, go ahead. Sorry.
String, can we make it not be the little chunks? And is there a way to get it to where it is like a string?
Like take it out and spread it out on the desk and stuff, like what you were saying? Or
yeah, because right now it's just like little pieces. Or is that supposed to be?
We can try. I'm game. Let me get some DNA out.
Yeah,
and if you have a glass stirring rod, sometimes that can, like you were saying, you saw it that way before, but that can help to kind of pull it out because the glass has a little bit of charge.
a lot of times it like clumps up into like a big long string more than mine did this time anyway. Mine kind of stayed as little white pieces sort of this time. And that's kind of unusual because usually it's kind of, more like a stringy,
right,
slimy white string sort of. So this one was a lot different than other ones that I've done. So,
okay.
Yeah.
That's what I was asking, because this is, like you said, this one's different than the one I'd seen before.
Yeah, me too. Yep. And not quite as much as normal, I think, or whatever.
And I would definitely recommend using the test tubes for your students instead of the little bitty tiny little things that we got, and then letting them transfer it over, because that way it's easier to pour, they can see it better. Like, I like the test tube model because the materials are so inexpensive that you can afford to do big amounts.
So I don't know if you guys can see the wads with the glare on my desk, but I think it was my second trial today. So it's been sitting for a lot longer. Like, it's a little bit more thick, like I think what you guys normally feel like. It still doesn't feel as thick as like a strawberry or banana compared to the one that I just did, putting it out on the desk. So
would like letting it sit to settle, does that help it? Does that make it easier? Does it make it worse? Or, if we were to put it in a centrifuge or something like that, would that help?
Yeah, the longer they sit, like the more kind of like balled up it becomes in the alcohol layer. But if you wanted to take it out and centrifuge it to the bottom into a pellet, I'm not sure. I mean, I think it it would get it all down there. But when you pulled it out, I don't know.
Was the shaking, do you think the shaking is maybe, which I didn't shake it hard, I just tilted it. Is that something that would make it break up?
It might. I did not shake mine in the test tube, and the most recent one was still it's still kind of breaking apart. But I think it would still be neat if you could put some kind of like stain on it and look at it under a microscope. It was really cool, and the kids would really like it. So Rachel, you're gonna do it tomorrow?
I've got to go find wheat germ.
Or strawberries.
Or okay, so do you have to mash the strawberries?
So get yourself some frozen strawberries, and then they can, if you have access to little baggies, they can put one in a little baggie and get it all smushy, 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 and then add the tenderizer or the salt and the shampoo or dish detergent. Dish soap works as well, and they can do it all right there, and then they pour the contents of it through, like I've seen them do it like through some cheesecloth into a test tube, right? Or you just pour it all into a test tube and then add the cold alcohol to the top, like, pour it down the side so it stays on top, and then it'll come right up to the top. And then no muss, no fuss. You just take those bags, and hopefully you take those bags and get them to rinse them out, recycle them at your nearest grocery store. Don't throw them away, right? Make them wash them out and turn them inside out, and use them for the next class. As long as they're not poking holes in them. That's what I would do.
Yeah, and we have poked holes before, like if you cut a little, like one of the little bottom corners off, and then just kind of like the juice, the stuff that you pipetted off goes into the test tube, and then you can put the alcohol, but then you can't reuse them. We need plastic baggies that are made from Danimer Science.
Yeah, that
Yes.
was it PHA? I'm gonna go get some of those straws so that we can try that.
I know. I wonder, I was wondering about the bacteria he was talking about. I have a lot of questions about the bacteria, but I didn't want to ask.
They're proprietary, so some of it is secret.
Yeah. Well, I just wondered, like, so are just normal bacteria, like, did their bacteria break it down, or they just use their bacteria to make it, or?
Yeah, so he said that they synthesize PHA the same way that like our bodies kind of make fat.
That's right. That's right.
Yeah, so they store it as like an energy source.
But then any bacteria, any just common.
Not any, I think, but that specific one does.
But then any will break it down.
Mm-hmm.
Okay.
Right, yeah, anything can break it down. Fungi, bacteria.
Okay, well, I'm gonna try it. So y'all say hi to Tiny. He's being
hello.
Aww.
Not very, but very tiny. But he's my big baby, and I've been ignoring him. So now he's letting me know I need to pay attention to him.
Hello.
Well, Megan, is there anything else that you wanted to cover with this, or does anybody else have any questions?
Any more questions?
Any more ideas or brainstorming?
Well, and if you guys do the lab with your class, feel free to post it to the group me or send it out. Post it on our social media, tag us. We love to see that kind of thing.
Anything specific for that we need to do? Like if I do this tomorrow with my students, does that count as the implementing?
For the implementation, you're going to need to do a video, like a demo video. So I don't know if you're prepared to video yourself tomorrow. But all of that is in the, it's in the Google Drive with the implementation plan.
Okay.
So yeah, you could do it, or you can just wait to do it again in the fall.
Okay. Do I have to do it with wheat germ?
No, you can use strawberries. You can use whatever you want to extract DNA from.
You could get a couple things and let your kids pick.
I think that's what I'm going to do.
Or what Trisha was saying, do food from home, ask your kids to bring a small sample of something. And then like have them put them into groups and let them pick one thing that they want to bring in per group. So you're not trying to get 77 different pieces of food matter, you know. You could have
About half of us are virtual, so my classes are about half the size they usually are. So if this is the year to do anything, this is
yeah. And it's also a year for you to get started doing it and do it small. And then next year when we're back to, hi everybody, like we came back full time Monday. We've been hybrid, or yeah, we've been on a hybrid schedule for the last 2 months. And so yeah, it would be a good way for you to like dip your feet in, get used to doing it with a small group and then do it with a big group. So I think they'd really enjoy it. Like I may have to change my lesson plans and go in tomorrow and do some DNA with them. I'll have to think about it.
I hope we have school tomorrow because there was, we received a weather alert email about potentially strong storms in our area from 5 in the morning until 10 in the morning.
Yeah,
So
we're on a 2-hour delay. So we're going into delay.
It's supposed to get to us about 10 from 10 to 12 is what they said. So we'll be right in the middle of lunch.
Oh,
Well, you know, you're safer at school than you are at home for some people.
That's right. That's right.
Okay. Well, I'm excited. I'm glad we did this. You know, I was like, oh, I've done this, you know, a couple of times, but you guys have really inspired me to do some different things. This is what I like about us getting together and doing stuff is that, you know, I get inspired by y'all. So thank you.
well, thank you. And I'm just really thankful that everything worked. And I know I have this funny glare, but I'm very paranoid to touch anything on my computer or this document camera. I'm very paranoid after last week. So, because even when I was on with Kristen, the Monday we did another trial run and the camera froze partway through. And so I just haven't wanted to touch anything hardly. So,
So, okay, I have one more question. We had to soak, or we had to put the wheat germ in the hot water, but I don't have to do that with the strawberries or the bananas, right?
Nope.
yeah.
Because I think strawberry and bananas will be a little bit easier to find out here in the middle of nowhere than maybe wheat germ.
Yeah, and you can buy the off-brand frozen strawberries and then just don't put them in the freezer. Just let them defrost and, you know, use that. And I've had a lot of success. Anybody else had success with frozen strawberries?
I haven't tried frozen. I would just always stop at Kroger on the way to work and grab a thing of strawberries.
Yes. And this is the wheat germ. I don't know if you guys can, I'll risk the camera.
Yeah, there you go.
So this is December of 2017. And this one worked.
Untoasted wheat germ.
Well, and I
Anyway,
guess with the wheat germ, you can kind of think of it like oatmeal. So that's why you need to soak it in hot water for a while, get it mushy.
Oh, my room is a mess. So don't judge me right now. This is my desk.
Oh, whatever.
Megan, you have seen my room. It is worse right now. It's probably all-time high of stuff everywhere. So
that's when science is happening. So
That's right.
yeah, it's been a really fun week. So I really hope that we're here tomorrow because we're virtual Fridays. So
You too, Miss Linda.
Cool. Well, and if you guys don't have anything else, I can go over some of the prep for next week, which I'll send that out. I really apologize for sending it out Tuesday afternoon. I totally meant to do that Monday and just lost track of what day it was. So let's see. So for next week, we are going to do 2 labs. There won't be a speaker, but you do need a couple of things kind of on hand. So we're going to be doing 2 labs. One is chromatography, and then the other is gel electrophoresis, like Matt talked about earlier. So for the chromatography, you have a nice bag that has everything in there except for the isopropyl alcohol. So that same alcohol that you guys are using right now, you don't need it in the freezer next week. It can just be room temperature. So you can take it out and leave it until next week. You will need your graduated cylinder and beaker. For that one, you will be doing some dilutions of alcohol. So the procedure has you do, I think, a 5% and a 25.
Yes.
Yeah, that's right. So Trisha will walk you through doing that. I know that the lab protocol has you make 500 milliliters of each, which you will not need. So you guys will scale it down. On there, I said, please provide some sort of container to put the diluted alcohol in. So it can be a beaker with some plastic wrap on top of it, or I have a bunch of those baby food containers, like the plastic with the lids at home. I just use those whenever I do this, and then label one 5 and the other one 25. Just, you don't want the alcohol to evaporate before you get a chance to use it. You will need water. So if you have distilled water, that's great. If not, tap works okay. Hey, Britton's cat.
Aww.
I love seeing animals walk across. It's entertaining. And then, so the advanced prep for that will just be to prep the Kool-Aid. I would recommend starting with the purple. You guys have a green packet too that you could play with later, but they should separate out about the same way, and you should see the individual colors. On gel electrophoresis, you guys will be making your agarose together. So you have a little tube of powdered agarose, and then you'll just need to add that to some buffer that you guys will also dilute. So you will need some sort of bottle to store that buffer. It can even just be a disposable water bottle. So if you have a Dasani bottle or something hanging out, or a Danimer plastic bottle, you can just use that, anything with a lid that you can store it, and then just label it buffer for later, because you'll have a little extra. For the agarose, you will need to heat it up. So you can use either a microwave, or if you have a hot plate handy, just don't heat it all the way to boiling, or not super hot. Don't put the hot plate on high, but you do want it to boil for a little bit. Generally, when you're making them, you use it as kind of to kill off some stuff, but also to get the agarose fully dissolved in the water. So however you heat it. And then, yeah, the distilled water for the buffer, and then something to cut the gel. You can use your micropipetting card if you don't have anything else. But some plastic card, or I do it with a plastic knife or whatever butter knife that's sitting around, to make those slits in the gel. So, but there is no advanced preparation for that, because you will do it all with Trisha.
So next week, Megan, did you want to start? Do we want to start and make the gel first and then let it solidify?
So buffer gel, and then, yeah, so you can do like get the gel started and get it poured, and then go back to doing all the kind of going through the lab.
Okay. Yeah, I didn't know if you wanted us to pour the gel and then do like Kool-Aid chromatography and then come back to the gel when it's solidified, or
that'll give it a while. Yeah.
Okay. And so the only thing they need to make is the Kool-Aid. They don't need to make the alcohol, right?
Right. Yeah. So you guys will go through the dilutions together and do that together. Do
you guys have any other questions about next week or
Nope, not me.
Okay.
We use shampoo. Any soap works, right? Any soap. Okay.
Yep. But you now have a tiny tube that you are welcome to use, and that should last you the whole day.
I think that, I don't know if there's anything else.
I think as far as we're concerned, we don't have anything else today. So if you guys get to your session 3 prep and you have any questions, feel free to reach out. But
thank you guys so much.
I'll go ahead and send it out tomorrow, maybe, just to give you all a heads up, since there were a couple things to collect. And to make up for sending it 2 days late last time.
Have a good night, guys. Be safe.
Thanks, everybody.